Good afternoon. Thank you for giving us the opportunity to present Ketamine 2022. I'm Marco Papagallo, the Chief Medical Officer at Relmada. We're excited to share data on the current development of REL-1017, esmethadone hydrochloride, in our presentation titled REL-1017, an NMDAR receptor antagonist for the treatment of major depressive disorder. In this talk, we will discuss findings from our phase 2 clinical trial of REL-1017 for the treatment of major depressive disorder and provide our understanding of why and how REL-1017 is able to induce its antidepressant effect. Next slide. REL-1017 is a novel, low-potency, uncompetitive NMDAR receptor channel blocker currently in phase 3 clinical trials for the treatment of major depressive disorder. Next slide. In our phase 2 trial completed in 2019, REL-1017 showed rapid and robust efficacy and a favorable safety, tolerability, and pharmacokinetic profile. The data from this trial was recently published in the American Journal of Psychiatry. Next slide. In this study, REL-1017 was investigated as an adjunctive treatment in patients who were experiencing inadequate response to a standard antidepressant therapy. This was a 7-day inpatient, randomized, double-blind, placebo-controlled study comparing REL-1017 25 milligrams and 50 milligrams to placebo. Patients were dosed in the first 7 days, followed for efficacy up to 14 days, and then for safety up to day 21. The safer interview was utilized after the initial screening to ensure patients entering the trial met the criteria for major depressive disorder to confirm quality results. The MADRS at baseline for all subjects was 34, a score categorizing patients as moderately to severely depressed. Baseline characteristics were well balanced across all 3 arms. Next slide. As you can see from the graph, both 25 and 50 milligram doses achieved statistically significant improvements compared to placebo, starting at day 4, then sustaining to day 7, and up to day 14, 7 days after discontinuation of REL-1017 at day 7. Differences in MADRS at day 7 were minus 8.7 for the 25 milligrams and minus 7.2 for the 50 milligram dose. And at day 14, minus 9.4 for the 25 milligram dose and minus 10.4 for the 50 milligram dose. For comparison, a typical NSSRI has about 3-point MADRS difference versus placebo. Another way to appreciate the efficacy findings is to look at the effect size. Here is as D. The effect size for the REL-1017 in this trial ranged from 0.7 to 1 during day 4 to day 14. Put this into context with other traditional antidepressant trials where the effect size are modest at best, around 0.34. Currently approved treatments can sometimes take as long as 8 weeks to show the onset of efficacy, where with REL-1017 we still will see a very rapid onset of efficacy. Next slide. Other important measures of efficacy are response rate and remission rate. The response rate is defined as at or more than 50% MADRS improvement from baseline. And remission rate is MADRS score of 10 or less at the endpoint. So both the 25 and 50 milligram doses demonstrated significant improvements versus placebo at day 14 for the response and remission rates. Next slide. So as far as the REL-1017 safety, there were no treatment emergent adverse events that led to patients dropping out or treatment discontinuation. Adverse events were infrequent. There were no serious adverse events or adverse event of special interest. Of relevance, no dissociative, cyclotomimetic, or withdrawal effects, no euphoria, no respiratory depression were observed, and there were no instances of clinically meaningful QTc prolongation. Next slide. In addition to the safety findings from the phase 2 results, we recently completed 2 human abuse potential studies in recreational drug users, comparing 1017 to oxycodone, as shown on table 1 on the left-hand side, and to ketamine, as shown on table 2 on the right-hand side. A poster presenting these results are being shown at this conference. I'm here to provide a high-level overview of the key findings of these 2 studies. The primary study endpoint for both HAP studies was the drug liking, which at this moment, which is measured on a 0-100, a VAS scale, 0-100, bipolar, where 50 is neutral, 0 means means maximum dislike, and 100 is maximum liking. So the results of the REL-1017 versus oxycodone are shown here. We utilized three doses of REL-1017. The 25 milligrams, the 75 milligrams, and the 150 milligrams. The same 3 doses were utilized in the HAP study for ketamine. 25 milligrams is the therapeutic dose that we use in the phase 3 trial, and 75 milligrams 3 times the therapeutic dose. Also the loading dose on day 1. And 150 milligrams is the 6 times the therapeutic dose, and it's the maximum tolerated dose in phase 1 studies. We performed a completer analysis for the oxycodone. We had 47 completers, meaning that subjects who completed all the 5 treatments, and for the ketamine we had 51 completers, meaning that of subjects who completed all 6 treatments. For the ketamine, as you see, we also included dextromethorphan as an exploratory endpoint. For the doses, oxycodone was an oral dose of 40 milligrams, and ketamine was intravenous dose of 0.5 milligram kilo, given over 40 minutes. All the 3 REL-1017 doses were statistically significantly different and lower than oxycodone and ketamine, with a p-value of less than 0.001. And of note, the REL-1017 doses were statistically equivalent to placebo in both HAP studies for the oxycodone and the ketamine as well. So these results provide further support for our understanding of the safety profile of esmethadone or REL-1017. The drug did not show, in essence, meaningful abuse potential in both HAP studies. Next slide. So how and why does REL-1017 offer this unique property for the treatment of a major depressive disorder? Our preclinical work today assessed the effects of REL-1017 among other NMDA receptor antagonists, including dextromethorphan, memantine, ketamine, and MK801. Of interest, the work showed that REL-1017 has a preference for the N2D subtype of the receptor. And this might be an important novel variable in understanding its clinical action and its effects as an antidepressant. Next slide. So our hypothesis is that in major depressive disorder, the N2D containing NMDA receptors may become tonically and pathologically hyperactive. And their dysregulated state would lead to an intracellular calcium overload and neurotoxicity in the affected neuronal population, with impairment of the physiological homeostasis of synaptic proteins and BDNF synthesis, ultimately impacting on neuronal and memory plasticity in the brain of the patient. Tonically and pathologically hyperactive N2D subtype channels may be a novel target for NMDA antagonists by downregulating excessive calcium signaling and positively impacting on neuronal and memory plasticity in the depressed brain. Essentially, we can restore the brain's ability to make new vital connections and neuroplasticity. Next slide. So what REL-1017 does is to repair the N2D containing channels hyperactivity by blocking the excessive tonic calcium influx, rekindle transcription and production of synaptic proteins, synthesis of BDNF, and ultimately promote the necessary neuronal plasticity that leads to recovery from major depressive disorder. It's of relevance. Our phase 1 studies have shown the REL-1017 can rapidly increase BDNF serum levels. Of interest also is the REL-1017 has characteristics of low receptor affinity and high traffic, which means, in our opinion, it correlates with rapid antidepressant effects with no dissociative symptoms. In contrast, ketamine has high potency and high traffic with strong antidepressant effects, but also with dissociative symptoms. Next slide. This slide here helps visualize the activity of the brain before and after the use of REL-1017. Of interest, the N2D containing channels among all subtypes have the highest probability of open configuration arresting membrane potential. So they are more likely to be tonically open and allow excessive calcium influx. The N2D containing channels do exhibit a higher glutamate affinity, a lower protection from magnesium blockade than other subunit receptors. So after treatment with REL-1017, the hyperactive N2D containing receptors return to operate physiologically, arresting membrane potential. Tonic calcium influx is normalized. Chronic excitotoxicity reverses, neuronal plasticity resumes, and depression recovers. The brain is then able to carry on the necessary connections, enabling major depressive disorder symptoms to go into remission. It's important to note that the development of REL-1017 for the treatment of major depressive disorder will contribute to the growing understanding and body of literature where now the N2D subtype may have an impact on several neurological conditions through its ability to normalize synaptic plasticity. Next slide. In conclusion, REL-1017 is dextroisomer of methadone and a low-potency NMDA receptor antagonist. Preclinical data indicates a preferential action on the N2D subtypes, which may be the key pharmacological effect of the drug's ability to increase neuroplasticity and provide antidepressant effects. In our experience, REL-1017 does not induce dissociative effect, euphoria, or respiratory depression. It showed no meaningful abuse potential in the HAP studies conducted in recreational drug users of opioids and ketamine. So overall, we have a very favorable safety tolerability profile across all the phase 1-2 studies. As far as efficacy, we have seen a robust, rapid, sustained, statistically significant antidepressant effects on all tested scales. Of relevance, the REL-1017 is orally administered. It's once-a-day tablet. And finally, REL-1017 phase 3 clinical trials for the treatment of major depressive disorder are in progress. Next slide. So here are the list of references that we have used in creating our presentation and will make available to you for your perusal. Next slide. And lastly, thank you for listening to our presentation. Paolo and I will be available for Q&A for the next 10 minutes to answer all your questions you may have. We're looking forward to a good discussion. Thank you very much.